student, the urinary system is actually probably the most difficult of all systems in terms of histology. Okay? Because not only do you need to know the normal histology, but you need to go beyond that, and you have to understand the electronic microscopic view of the glomerulus, okay? So that you could understand how they affect various diseases. Okay? So I'm going to show you a few slides from my PowerPoints, but I won't show you my whole PowerPoint. I'll just show you the key slides. So we'll look at the anatomy of the kidney. So this is looking at a gross section of a kidney that has been dissected. Okay? So the outer portion here, this darker portion, is the cortex, while the inner portion here is the medulla. Okay? So then, if we look at it histologically, the difference is the cortex will actually contain the glomerulus, okay? Which I'll call from now on glom for short. Glomerulus, glom. Okay? Your medulla is going to contain mostly your loops of Henle and your collecting tubules. You will not see any of the glom in the medulla. Okay? So for the basic functional unit of the kidney, what's the basic functional unit? It is the nephron. That is correct. Now the nephron consists of two parts, the renal corpuscle and the tubules. Okay? The renal corpuscle is composed of the glom and the Bowman's capsule. Together they make up the renal corpuscles. The tubules consist of the proximal, the loop of Henle, the distal convoluted tubule, the collecting tubules, and ducts. Okay? And we have different types of nephrons, but then I want to show you the actual structure of a glomerulus. Okay, so of a glom. Let's go to this one. All right. So in the center, we see the glom. What is the glom actually composed of? What is it actually? Do you know? It's actually capillaries. Okay? It's a ball of capillaries that are supported by cells called mesangial cells and also podocytes. In fact, you can see some of these capillaries. The lumen has red blood cells in it. See? There's a red blood cell here. So those are the capillaries. Okay? Then for the capsule, the Bowman's capsule, so this is also the same Bowman that you saw in the Bowman's gland, in the olfactory epithelium, as well as, if you look at the eye, Bowman's membrane. Okay? So there are three things in histology named Bowman. Bowman's capsule, the Bowman membrane, and the Bowman glands. Okay? So here is the Bowman capsule. The Bowman's capsule is divided into two regions. You have the parietal layer, which is composed of simple squamous epithelium, and then you have the layer that's directly on top of the glom, which is called the visceral layer. The visceral layer is composed of a very important type of epithelial cell, which we call the podocyte. Okay, the podocyte. And then in between it, we have this space. This space is called the Bowman's space, or also known as the urinary space. Okay? This urinary space becomes continuous with that of the proximal convoluted tubules. Okay? So that's a structure of a glom. But it's actually not enough to know the histology. We need to understand it a little bit more in detail. Okay? We need to see, I'll show you this diagram. Okay? This is a diagram of a glom. Okay? The glom has two different poles. Where the blood vessels enter and exit, that's called the vascular pole of the glomerulus, or the glom. Where it's continuous with the proximal distal convoluted tubule, that's the urinary pole, or the tubular pole. Okay? And if we look here at the glom, we see these bright yellow cells. See these yellow cells? These yellow cells have little cytoplasmic extensions, which we call pedicels, okay? Or foot processes. And in between the foot processes, there's a little space. Okay? That space is called the fenestration slits. Okay? So I have another picture of the glom. Here's another picture. Okay? So you see the podocytes are here. These are the pedicels, and then they wrap around the capillary, and then they have little openings where you can see the part of the capillary. That's the fenestration slits. Okay? So the function of the glomerulus is actually going to be filtering the blood. Okay? So things that are in the capillaries will be filtered out and enter into the urinary space, where it will continue into the tubules. Okay? So knowing that, what are actually the physical barriers of filtration in the kidney? What do you think? Knowing that it has to pass through those structures, what are the barriers that they have to pass? Okay. Basement membrane, correct? So what type of capillary is seen in the glom? Yes, it's a fenestrated type of capillary. What does it mean when a capillary is fenestrated? Meaning they have little holes or little openings. They're fenestrated. Okay? So we have here a diagram. Okay? And then I'll show you what it actually looks like on the electron microscope. Okay? So here we have the capillary lumen. This is the capillary lumen. So blood is here, then the urinary space would be here. Okay? So in order for something to exit the capillary, it must pass first through the endothelial lining of the capillary, through the fenestrations that you see here. That's the first barrier, the capillary endothelium. The second barrier is known as the glomerular basement membrane, or GBM for short, glomerular basement membrane. Okay? I'll show you what it looks like in the electron microscope a little bit later. Okay? And then finally, you remember you have podocytes that are wrapping their foot processes around the capillary. They have to go, the particles or substances that are being filtered out will have to travel in between the slit diaphragm, or the fenestration slits. Okay? So again, the barriers of fenestrations are the capillary endothelium, the glomerular basement membrane, and the fenestration slits. Okay? In order for substances to move out of the capillary and enter into the urinary space. Those are the barriers of filtration. Let me show you now what it looks like on an electron microscope. And I will be asking you to know this because we have a PowerPoint exam, and I can include any picture I find on those exams. So let's look at it a little bit in high power first, because I think it's easier in high power. So this is the electron microscopic view of the glomerulus. So here, this is the capillary lumen. Okay? This is where the blood would be. Here is the endothelium. Simple squamous endothelium. Okay? You can see there are little openings, which are the fenestrae. Okay? That's the fenestrae. Here now is the GBM. Okay? In actuality, the GBM is divided into 3 layers. Okay? There are 3 layers. Okay? In the middle is this very dark layer. You see this dark band? That's called the lamina densa. Okay? The lamina densa. And it's sandwiched between 2 light areas. These light areas are respectively called the lamina rara interna and the lamina rara externa. Okay? Okay? So when you see these, the internal refers to the endothelial surface. The external refers to the podocyte surface. Okay. When you go to pathology, they're going to use different terminology sometimes. They're going to call this region as subendothelial area. Okay. And then they'll call this region up here subepithelial area region. Okay? Just don't be confused with the terminologies. Okay? It's called endo because this is the endothelium. Epi, modified epithelial cells, the podocytes. Okay? It's important to know that because when you go to diseases of the kidney, particularly glomerulonephritis, one of the things that they use is the electron microscope. Okay? And when they look at the basement membrane, sometimes they're going to find immune deposits. Okay? And depending on the location of the immune deposits, is it subendothelial, subepithelial, you can come up with various diagnoses based on the pattern and the location and distribution of those immune complexes. Okay? So you do have to know what the GBM looks like on electron microscopic view. Okay? But it's not that difficult. Just know the barriers. Endothelium, GBM, your fenestration slits. Let's keep it simple for that. It can go a little bit more further in detail, but for now, let's keep it simple. Okay? So that is the barriers of filtrations. Questions so far for the GBM? Okay. Now there's another cell that's also important. We talked about the capillary. Here is another diagram. We talked about here, this is the capillary endothelium. This dark purple line here is the GBM. And then see the light cyan color cells here? These are the podocytes. Okay? And these here are the slit diaphragms and the fenestrations. Okay? Are here, the fenestrations. Okay? But there's another cell in the glom. We have these violet cells or pinks. These little light purple cells. Hello? These light purple cells here. These light purple cells are called mesangial cells. Okay? The light purple cells you see, is it purple or is it pink? Okay. These cells are the mesangial cells. There are two types of mesangial cells. You have intraglomerular mesangial cells that are within the glomerus, and you have mesangial cells that are outside the glomerus, which is called the extraglomerular mesangial cells. The extraglomerular mesangial cells are also known as your Lassus cells. Lassus cells. So these mesangial cells, their main function is actually for structural support of the glomerus. They're the ones holding the capillaries together. If they weren't there, the capillaries would float away and just be floating around inside the renal corpuscles. So the main function is for structure. They do have other functions, like ingesting proteins and debris, phagocytosis, but those are secondary. It will suffice that they are support. Okay? So here is the filtration barrier. You have, again, capillary endothelium, fenestrations, GBM, and then finally, from the urinary space, it enters the proximal tubule. Okay? All right. Now, there's one last thing that we'll talk about the glom. There's another structure next to the glom. And this structure is called the juxtaglomerular apparatus. The word juxta means next to or adjacent to. So this is a structure next to the glomerus. It is composed of three components. Okay? The first one is something you can actually see in the light microscope. That's called the macula densa. Okay? The macula densa. To look for the macula densa, you need to look near the vascular pole of the renal glomerus and look at the distal convoluted tubule. Within the distal convoluted tubule, you'll see a cluster of very tightly packed cells that are a little bit taller than normal. That's the macula densa here, this little pink one. And I'll show you a real picture later. Now the function of the macula densa is it detects sodium levels within the distal convoluted tubule. Okay? It's detection of sodium levels. The next cell, it's the full terminology of the next cell is called the juxtaglomerular granular cells, or the JG cells. Okay? Here they call them granular cells, but the full name, juxtaglomerular granular cells, JG cells. Okay? These are modified smooth muscle cells that contain granules. Do you know what granules are, or what's being stored in the granules? What do you think is being stored there? That's correct. It's renin. Renin. You will talk about renin in your physiology class. It's very important. It's one of the most important things you'll have to know because it's very important for regulating blood pressure. Okay? But let's not focus on that because I could spend a long time talking about that. So just know they're granular cells. They're a little difficult to look in the lab. Okay? Usually they use electron microscopes to find it because they're a little bit challenging because they're jumbled together with your extraglomerular mesangial cells. So I won't ask you to identify it in the microscope, but I might as well show you a diagram like this on your PowerPoint, so you still do have to kind of know it. Okay? That's why in histo you don't just have a moving exam, you also have a chrome exam. Okay? All right. Now the last cell is your extraglomerular mesangial cells, or your Lassus cells. Those are the purple. Again, they have the same function as the intraglomerular mesangial cells. They support the glomerular apparatus. They're the ones holding it together. Okay? All right. So let's skip through some more slides before I show you the actual slides. I want to show you some other things that are not quite easy on the lab. Here. See this? The arrow? That's the macula densa. You see that tightly packed cluster of cells near the vascular pole of the glom in the distal convoluted tubule? That's your macula densa. Okay? Here's another picture. That one cluster of cells in the distal convoluted tubule. Okay? That's the macula densa. That one is you can find it. It might take a while because it has to be cut perfectly in the middle. But if you spend some time, you will be able to find it. Okay? All right. Now I do want to show you another thing that's not so easy to see. Okay. Here we're looking at a medullary ray, or you can look at it, the medulla. This is a collecting duct, the one that's being pointed with blue. Okay, the collecting duct. Okay. The collecting duct and the collecting tubules are the very last portions of the nephron. Okay? This is the last portion where urine can be modified. Okay? Now there are two cells within the collecting ducts and tubules. The majority of these cells are the paler cells, the ones that are being pointed by the orange arrow. Those pale cells are called principal cells. Principal cells. They're the most common cells. Then the darker redder cells that are highlighted with the red arrow are called intercalated cells. Okay? Now, what's the function of principal cells? Anyone? Didn't read in advance? Principal cells contain aquaporins. Yes. What are aquaporins? Yes, they're for water transport. So when not activated, these aquaporins are just being stored in the cytoplasm. But in response to a certain hormone, the aquaporins, which are normally stored in the cytoplasm of the principal cells, will be moved into the apex of the cell. Therefore, incorporated in the membrane so that what would happen to water? Water would go in. Water would go in. So, what is the hormone that's responsible for the movement and incorporation of aquaporins into the apex of the cell? What is that hormone? ADH, antidiuretic hormone. That's secreted in what part of the body secretes ADH? Posterior pituitary. Which is stored in what structure? Do you remember? Herring bodies. Do you still remember your histology? Everything falls into place. So, aquaporins ADH. Okay? So if there is an antidiuretic hormone, what would happen to the urine? Would it become diluted or will it become concentrated? It's concentrated because we are taking out the water. So if we are taking out the water, there is concentrated. All right. And for the last cell we have here, the intercalated cells. Technically, there are two types of intercalated cells, but I will not ask you for the difference or to identify them. Just call them intercalated cells. They are for acid-base bonds. Acids and bicarbonate. Right. And then one thing I do want to show you, okay, we have the male urethra. Okay? The male urethra is divided into three portions: the prostatic, the membranous, and the penile urethra. Okay? So I was going through the lab. We do have a histology of the prostate gland, but unfortunately most of the slides do not actually have the prostatic urethra. Okay? The prostate is a relatively big gland. It's a big organ. So if you don't cut it directly where the urethra is, you might not have it. Okay? So I will not ask you to identify it really in the lab, but do know it just in case. Okay? Do know it. Okay? I will show it to you though. Here it is. These are the glands of the pancreas, of the prostate. Okay? They have a very unique undulating morphology, which I'll show you in the later slide. And ideally, this is what you should have been looking for, the urethra. Okay? It's also lined by transitional epithelium. We don't have that in our slides. But it's okay though. Don't struggle. Here's the transitional epithelium. You already know that. Yeah, transitional. This is the penile urethra. Unfortunately, you don't have this slide. I'm surprised. I thought I had this slide when I was in med school in here. I thought we had a slide on that. So this is a cross-section of the penis. Now the penis has three bundles of erectile tissues. Two of them are on the dorsal portion and they're called the corpus cavernosum. There's two. On the ventral surface, you have the corpus spongiosum. This corpus spongiosum expands at the end and becomes the glans penis or the head zone. But what's important there is this is where you're going to find the penile urethra, in the ventral portion of the corpus spongiosum. Okay? What we actually have in the lab, which is pretty interesting, is we have the female urethra. So the female urethra is only one segment. It's not divided into several segments. So only one segment. And it's lined by transitional epithelium, except at the very distal portion where it becomes similar to the epithelium of the vulva, which is skin, stratified squamous. Okay? So let's now look at the actual slides. Yeah. Let's look at the actual slides and see how much we can identify. All right, let's start off with this organ here. So we see a capsule. So this is the capsule. What organ do you think this is? All right, that's the kidney. What region do you think this is? Cortex, medulla. We see the presence of our glom. Don't use glom for the exam, please. You have to write it out. I just say glom because it's very cumbersome to keep saying glomerous. So this is the cortex. Over here is the medulla. Okay? So let's focus on the cortex. And we also see something else. Let's see if it's here. Maybe it wasn't this slide. Okay, it's not this line. These are the medullary rays here. They're extensions of tubules that will go through the cortex and into the medulla. You know, you don't need to know that. Let's go to the cortex and let's look at a nicely stained area. All right. And let's go to high power a bit. All right. This is kind of shaped funny. Let's look at a better one. This one. So this structure is the, yes, glom. So what would be lining the glom? That's the visceral layer of the Bowman's capsule that are lined by what cells? Podocytes. Okay. Now, I'm not going to ask you to differentiate which one is the podocyte here, which one's the mesangial cell, which one's the capillary endothelium. That's a little bit advanced for you. So just know the visceral layer are podocytes. Okay? But those questions are possible for side questions. Okay? How about here? What's this layer of cells here? So this is the parietal layer of the Bowman's capsule, and these are simple squamous cells. And then what about the space? This is the urinary space or Bowman space. Both are correct. Okay? Now let's move around a bit and see if we could find some things that are, if we are lucky, if we can find a macula densa, a very ideal macula densa. Not so ideal. Let's see. Well, it's okay. May have to take a while to look for the macula densa. Oh, but we do have something here. We got an interesting section. Here's the urinary space, right? And see, it's going to continue into the tube here. There's a tubular pole. That's the tubular pole. All right, but it's okay. Let's continue on. Most of the tubules that you see around here, this is the proximal convoluted tubule, these ones here. The proximal convoluted tubules are lined by simple cuboidal epithelium with microvilli. Although the microvilli tends to be very fragile, and sometimes it breaks off. See, here it's breaking off, and they're forming, it looks like particles there. Okay. These are the microvilli here, and then there's some broken off here. Okay. So this is the proximal convoluted tubules. They also appear to be a little bit more red in color because they have a lot of mitochondria, which is needed for the absorption and secretion of various particles within the tubules. Now let's compare it, with this tubule here. This is a distal convoluted tubule. Okay? Distal convoluted tubules, they tend to be short. Okay? Notice the thickness of the proximal compared to the distal. The distal is short. Then they tend to be a little bit more pale. They don't have that red, nice red color that you see in the proximal. And also, they have very scanty, if ever any, microvilli. Okay? So this is the distal convoluted tubule. This is the proximal convoluted tubule. Okay? All right. So what we have here. So let's focus now on the medulla. Okay. So the medulla are composed mostly of your loops of Henle and your collecting tubules. Okay? These thicker ones, these bigger tubules are your collecting tubules. Okay? Smaller ones are your loops of Henle. The loops of Henle are divided into your thick and thin segments. Okay? If it's thin, it's going to be more squamous, like this one, squamous epithelium. Thicker limbs are cuboidal. Okay? And then for the collecting ducts, these are the biggest ones here. Okay? And in the medulla, most of the time they are lined by these very pale cells, which are their principal cells. These are your principal cells. Okay? The intercalated cells, you can sometimes see them here, but they're scattered. Okay? And you may want to focus in the medullary ray. Let's see if we could look at a medullary ray. Here it is. Oops, sorry. You see this dark red cell next to this pale cell? So this dark red cell is the intercalated cell. And then this light cell is the principal cell. Actually, this is a good slide. You see two of them. Two of them. Okay. All right, so this is your collecting duct or tubule. Okay. All right. So those are the cells of the kidney. All right. Any questions so far? Questions? Huh? All right. So try to find all the structures as possible. Okay. Let's move on. Next, we have another organ here. We see that it has a stellate-shaped lumen. Okay, we see a stellate-shaped lumen. If we look at the lining epithelium, what's this epithelium? It's transitional or urothelium. Okay, it's not squamous. Stratified squamous usually has an undulating basement membrane. This is smooth. Okay, this is also relatively thicker than your typical stratified squamous, except for keratinized stratified squamous in your thick skin. So this here is an organ with a stellate lumen, and then it has transitional epithelium. Then we see a lamina propria, and then we see a very thick muscular layer, and surrounding that is your adventitia. So what organ do you think this is? This is the ureter. Okay? So this is the ureter. Now, the muscular layer is a little bit different, okay? Because the way they're arranged is different than what you're normally used to. In the digestive tract, how many layers are there in the muscular layer?
4.
How many layers usually for the standard? 2. And what's the arrangement in the digestive? Inner circular, outer longitudinal. But in the ureter, it's usually 2 layers, except they're backwards. In this time, what's the inner layer is going to be longitudinal, and it's the outer layer that's circular. Okay, it's backwards. But even more confusing is the very distal portion of the ureter would actually have 3 layers. There's an additional outer longitudinal layer. So it goes inner, longitudinal, then it goes middle, circular, and then outer longitudinal again for the distal portion. Okay? So just take note that the muscular layers are arranged opposite to that of the digestive tract. Okay? So this is the ureter. What's this one? Oh, this one is actually the prostate. I was trying to see if we had any of the prostatic urethra, but I don't think our slides have it. But just in advance for the male reproductive system, the prostate consists of glands. See? These are the glands, and they have an undulating pattern. See? They're kind of undulating. Okay? And then the surrounding stroma is fibromuscular stroma. Okay? So this is a typical prostate. Undulating glands, fibromuscular stroma. Unfortunately, there is no prosthetic urethra in this one. So you don't have to draw it. But that's a little sample for male reproductive system. What we do have is this. And I know the staining is not ideal, but the staining is transitional epithelium. Okay? And we see a prominent lamina propria with a lot of blood vessels. Okay? And the muscular layer, which is not quite as prominent. This is the female urethra. Okay? This is the female urethra. Okay? Then for the last structure, upside down. So staining again is not quite ideal, but let's look at the epithelium. Notice it has this prominent stripe on the surface of the epithelium. What's that? You know what this stripe is? It's the condensed border. That's correct. This is the condensed border. So what type of epithelium are we looking at? Transitional. Do you think this is a full or empty urinary bladder? Do you think? Based on the appearance of the epithelium?
Empty.
Full or empty?
Empty. Empty.
Oh, you seem to be, you have different opinions. Okay. If this was an empty urinary bladder, you would see more of the dome-shaped cells. Okay? But what's happening is urine is pushing up against it. So the epithelium looks a little thinner, right? It's thinner, you don't see the dome cells. Okay? Let's compare it to this one. Back to this previous slide that I showed. Look at the thickness. Look at the thickness. Okay? And I know the epithelium is chop. But there would be some dome cells. See it projecting here? A little dome cell. Although it's a little chop, but you can see little dome cells. So this is the full urinary bladder, or dilated, distended. Okay. Then the other thing you need to know about the muscular layer, what type of muscle is this? Skeletal, smooth, or cardiac? It's smooth muscle. So what's the name of this muscle that would be here?
Detrusor.
It's a detrusor muscle, right? So during urination, this involuntary muscle will squeeze the urinary bladder, okay? All right. So that's the histology of the urinary bladder. And the outer would be either serosa or adventitia based on its location. Okay? All right, so those are the slides for the urinary system. Are there any questions? Questions?